Hello, everyone. One of the things I've noticed over the last couple of years is how poor the general level of understanding is in the population with regards to immunology, virology, vaccinology, cellular biology, these kinds of things, right? And it just so happens that these were the units that I studied for my undergraduate course in biology at university. And so, given that I am a teacher and that this is what I do, I thought it would be very helpful to create a video or maybe a series of videos, I'm not sure yet, that explains this to the layperson in such a way that they can understand more fully the conversations that are going on around them and be better informed in terms of their choice making. So what we're going to need to do is we're going to need to understand firstly, how do viruses infect cells and what do they do having infected the cells? Secondly, how does the immune system work to stop viruses infecting cells in the first place? And what happens once a cell has been infected? What does the immune system do about that? Then we need to go into a little bit about the cell itself, right? Some of the fine structure of the cell and some of the internal workings of the cell. In particular, we need to understand how proteins are synthesized by the cell. So we're going to need to understand how DNA codes for proteins and how DNA transmits that information out into the main body of the cell where the proteins are made. So some of this is going to be pretty high level stuff, right? This is going to be undergraduate, university level stuff. Some of it doesn't need to be as deep as that. So what I'm hoping to do here is tailor these videos specifically to give enough depth to understand properly and disregard all extraneous detail that doesn't actually help to inform this. Yeah, so this is a necessary starting point. I mean, there's actually some debate as to whether viruses are actually organisms or not, because they don't really do anything other than reproduce. But let's take a look at a diagram of a sort of generalized virus. And these are the key features that we need to understand. So the virus is encapsulated in a protein shell. And on the surface of that protein shell are bits of protein sticking out. These things are called antigens. In the center of the virus is a piece of genetic material. There are RNA viruses and DNA viruses. So it can be either DNA or RNA in the virus, and the actions of the two are slightly different, but it doesn't really matter. The point is that when the virus infects the cell, once the virus has entered the cell, the virus's genetic material gets incorporated into the genetic material of the cell, and it reprograms the cell to make more viruses. Viruses come in a wide variety of shapes and sizes, but they broadly break down into four different classes. And here's a little diagram showing those. And I've got some electron micrograph images of some specific viruses to show you just very quickly. So this one here is called a bacteriophage, so called because it infects bacterial cells. And you can see it looks like a kind of injection system, and that's exactly what it is. It lands on the surface of the cell and it injects its piece of genetic material into the cell. This is Ebola. This is a particularly messy, hairy looking virus, and a sort of generalized round virus with nubbins on, like the one in the diagram. And this is a blown up version of the antigens on the coat. This is just the bits sticking out of the surface of the cell. So this is unbelievably high magnification because viruses are very, very, very tiny things. So really what we need to understand about viruses is that they are protein shells with a piece of genetic material inside them, and that once they've entered the cell, then the genetic material reprograms the cell to construct new virus parts. This animation explains this pretty well. So what we're doing right now is sort of zooming over the surface of the cell, and this black dot coming into view is a virus seeking to infect this cell. And so you can see on the surface of the cell these proteins sticking out, these antigens sticking out, and the virus has on its surface antigens sticking out as well. And so the reason that not all viruses infect all things is that each species has its own very particular configuration of proteins on its cell surface, and the virus particle has to match up with those in order to enter into it. And so, in order for a virus to cross over from one species to another, it has to reconfigure its own protein code. But when the protein molecules when the surface proteins on the outside of the virus and the surface proteins on the outside of the cell are of the same configuration, then the virus is able to trick the cell into letting it in. We'll go into the cell membrane a little bit more, but it's kind of fluid in nature. And when it does, when it matches up surface protein with surface protein, it tricks the cell into saying it's okay to let me in, and the cell does so. It just lets it in. That is now an infected cell. Once the virus has entered the cell, the immune system can't get it anymore, and that is now an infected cell. It's important to understand that once a cell has been infected, it is lost to the body. The only thing we can do now is to destroy that cell, and so that's what the immune system does. It recognizes the cell as not self anymore, and it goes ahead and destroys it. This self, not self, is right at the heart of how the immune system works. In fact, autoimmune diseases are the body mistakenly thinking that self is not self and goes ahead and attacks it. On the surface of the cell, on the membrane of the cell, are expressed protein shapes. The immune system recognizes those protein shapes as either being self or not self. Once a cell has been infected and it started to produce viral particles, then those can be seen by the immune system on the surface of the cell, and it recognizes that cell as having been hijacked. It is no longer self and must be destroyed. And here is a very cool video of a T cell destroying an infected cell. You can see it recognizes it, it approaches it, it wraps itself around it, and it denatures it, destroys it, stops it becoming a threat to any other cells. So you can see that allowing cells to become infected means having to destroy those cells, and this is quite a high cost for the body to have to pay. It's much better if you can stop cells getting infected in the first place, and this is really what the immune system is all about. And so the immune system can be thought of as existing in two parts, one called the innate immune system, the other called the adaptive immune system. So the innate immune system is non-specific, right? It simply recognizes things which are not self. They express on their surface antigens which are not of the body and therefore should be destroyed. And this is a macrophage. What it does is it gobbles things up. It just reaches out and gobbles them up. And when it does so, it releases these things called cytokines. Cytokines stimulate the release of these things called neutrophils. And then these neutrophils and killer cells essentially go on the rampage and just destroy everything which is not self that they find in their path, including infected cells, right? So infected cells and foreign body particles are simply destroyed. Now, the debris left behind by this destruction is picked up by these things, dendritic cells. They wander around the place, and viral particles stick to them. They wander off, they pass on the little bits that they've collected, right? Here are little bits of virus. Here's what this little piece of virus looks like. That information is transmitted to B cells, and those B cells produce antibodies which are designed specifically to fit to the antigens on the surface of the virus. And once they have done so, once they have stuck to the surface of the virus, those antigens no longer work to unlock cell membranes, right? Those viruses, once they've got antibodies stuck to them, those viruses can no longer enter cells. They no longer pose a threat. So just to go over the B cell mechanism again, because it's worth remembering, let's start with the virus, which gets obliterated by a killer cell, and the pieces from the outside of the virus, those antigens, come into contact with a B cell transported by a dendritic cell or just by virtue of coming into contact with each other in the blood. You'll see there that B cell is called a naive lymphocyte. Naive meaning it has not yet been programmed to make any particular type of thing. It is awaiting programming is kind of what that naive means. And so once it's exposed to that antigen, once it comes into contact with that antigen, this activates the B cell and it grows into a lymphoblast. A lymphoblast is a cell ready to divide, and you can see there it divides in two, one of which becomes an active B cell producing antibodies specific to that antigen, the other one of which is a not active now but ready to become active in the future memory cell. And so months, years, decades after the infection has passed and we're no longer producing antibodies to something, we still have the memory cells floating around. And if that virus enters the body again, those memory cells know how to produce those antibodies. Don't need to go through this whole process anymore. The memory cells will become active very, very quickly, start producing antibodies almost immediately, inactivating the virus, preventing the virus from infecting the body. This, broadly speaking, is how long-term immunity works. We don't have all the antibodies to all of the infections we've ever encountered floating around in our system. That would give us blood clots. Antibodies are proteins. If we had all that protein floating around in our blood, we'd be getting blood clots all the time. And so what our immune system does is it stores the recipe for the antibodies in these memory cells. So just to return to this diagram in order to sum up, you'll notice that there are some cell types on this diagram which I haven't mentioned, and I have given a simplified, stripped down version of the story. It's true. However, the basic point that the innate immune system is non-specific and simply destroys anything which is not self, and that the remnants of that are picked up by the adaptive immune system in order to create bespoke, tailored responses to prevent cells from getting infected in the first place. That is absolutely the case. Once we understand how the innate and adaptive immune systems work, then it's pretty easy to understand how traditional vaccines work. As you can see on this diagram, it's this very, very simplified diagram, but it's good enough. It's good enough for the purposes of our understanding here. An attenuated form of the virus is injected into the body. By attenuated means it doesn't actually enter and infect cells. So it looks like the virus, but it doesn't act like the virus. But it looks sufficiently like the virus that the innate immune system recognizes it as not self, attacks it, breaks it up, exposes it to the B cells, which can create specific antibodies to it, all without the body actually having to get infected, without cells having to actually be destroyed. It's really important to understand that the immune response takes place outside the cell. Once the virus has entered the cell is dead to the body. And so the goal of the immune response is to stop things getting into the cell in the first place. The innate immune system is messy. Vaccination is a way of training the immune system without having to go through actually getting infected and having cells lost. Traditional vaccines are, in principle, a way of stimulating the immune system to do what it does anyway, without introducing any foreign bodies into the system at all. I mean, obviously, there are adjuvants and preservatives involved in vaccine production, and so it's not quite as clean as that. But in principle, traditional vaccination doesn't require the body to do anything that it doesn't already do. And therefore, the chances of collateral damage from traditional types of vaccination are very, very low. So just a couple of final thoughts before ending this part. Firstly, both the immune system and traditional vaccinations, because they're essentially the same mechanism, work best against stable viruses. By stable viruses, I mean ones that don't change very much over time. And so things like measles and chickenpox and mumps and whooping cough, these kinds of things, exposure to the pathogen itself or vaccination are very effective because the virus doesn't change much over time. And so the memory cells record an excellent image of the antigens and are able to make antibodies very quickly. However, of course, it's not that simple. Many viruses change their form. In flu, for example, there are up to a dozen flu strains circulating at any one time, and they mutate and change pretty frequently. And so, we don't just get flu once and then never get it again. We get different flus, and we get them repeatedly. And so, there's an arms race going on between the immune system and the virus world, right? Viruses update their antigen codes to slip past the immune system. The immune system updates itself to take account of the new strains, and so on and so on. And this goes on all the time. The other thing that I think is really important to understand is that when we get symptoms, when we get a fever, when we get mucus and coughing and sneezing and all of that kind of thing, it's not actually the virus which is causing these symptoms. It is our immune response to the virus which causes these symptoms. So we raise our temperature because it creates an inhospitable environment for the pathogen. We produce mucus and cough and sneeze in order to trap particles of the pathogen and get them out of the body, right? So all of these symptoms are actually our own body's response to the pathogen, not the pathogen itself. So all that said, I'm going to stop here and I'm going to do a part 2 where we're going to go inside the cell. Up to now, it's been pretty straightforward. Everything we've been talking about, or mostly anyway, has been taking place outside the cell. But in order to understand the mechanism of action of the novel vaccines, we need to go inside the cell because these vaccines do that. They go inside the cell very much like a virus does, and they reprogram the cell. And so in order to understand their mechanism of action properly, we need to understand how the cell membrane works. We need to understand how DNA is stored in the nucleus and how the information contained in that DNA is transmitted out into the body of the cell for proteins to be manufactured. And so that's all going to be in part 2. It's going to be a little bit more quite a lot more in depth part 2 than this part has been. But I hope you have found this informative, and I look forward to seeing you for part 2. Thanks very much for watching.